Now that we've gone through the changes that occur in the neuron as ions flux back and forth the membrane as part of our transport that we observed with the voltage-gated channels and chemically gated channels, we have to look at how that same electrical activity now can be extended down the length of an axon. And then once it gets to the length of the axon, how does it release neurotransmitters to either evoke a response in a second neuron or an end organ? So we're going to have to understand how that action potential spreads along an axon. And that's going to be by looking at the actions of the voltage-gated channels along the way, the nature on how it can go from point A to point B, either by saltatory or continuous conduction. And we're going to look at the factors that encourage one directional flow of this information. And that's going to include the refractory periods. So far, we've looked at an awful lot of electrical change in the membrane of a neuron. We've looked at action potentials, chemically gated channels causing greater potentials, and we kind of got right up to the actions at the trigger zone. In the last section, we saw how greater potentials in the dendrites and the cell body can bring about an action potential in that very first pair of voltage-gated channels at the beginning of the axon or at the trigger zone. Now we have to take that one step further on how that action potential can continue on down the neuron. So when we go back and look at the pairs of voltage-gated channels that are present here, sodium versus potassium, and both of them taking part in that typical action potential pattern, steep depolarization, steep repolarization, a hyperpolarization period, and then back to rest. We will see that we get the opening of our sodium voltage-gated channels first. When we hit minus 55 millivolts as a threshold, sodium rushes in. That's going to cause our sharp depolarization period up until we get to plus 30. At plus 30, the sodium channels close and become inactive, while the potassium channels now open up. The potassium channels that are in pairs with these sodium channels open. The sodium channels open first for our depolarization phase. Subsequently, the potassium channels open up as our repolarization phase. So every time we get a sodium channel open, its partner potassium channel would automatically open. And the ions that flux across the membrane as part of both of these channels taking part will give us our full action potential cycle. But when we look further down the axon, we will see additional pairs of voltage-gated channels. One sodium, one potassium, as we look down the length of the axon. So again, when the first sodium channel opens, it automatically opens up its partner potassium, because it's the sodium rushing in that hits the threshold for the potassium voltage-gated channels. But as this sodium opens up, we also see the very next sodium open up. So every time a sodium channel opens, it's going to turn around and open up two other channels, its partner potassium and the very next sodium in line. And that's how we start to get this propagation or transmission down the axon. Potassium rushes out. At the same time, sodium is rushing in the next pair, which of course, now that this sodium has opened up, it's going to open up its partner potassium as well as the next sodium channel in line. So now when this sodium channel opens, it's going to open up this potassium and this sodium. And by doing that, we go step by step by step the length of that particular axon. As long as we get this very first pair of voltage-gated channels open, the rest of these will happen automatically. Think about these like dominoes. We've all seen those arrangements of dominoes that are stood up on end. You knock down the first one, it knocks down the second, which knocks down the third, which knocks down the fourth. The only part of that sequence of events that needed your input was the first one. The rest of it happened automatically. That's what we see with this continued propagation. Now, this will only occur if these pairs of voltage-gated channels are close enough together. Because, of course, any sodium that enters the cell here, of course, is going to be brought back outside the cell to help reset our resting membrane potential. So we need to have these pairs of voltage-gated channels close enough together so that when we get an action potential arise from one pair of voltage-gated channels, it's going to have a big enough effect downstream on that axon so we can still at least meet the membrane potential threshold for the next sodium channel. I've often called this our circumference of influence. When we open up one sodium channel and we see that sharp depolarization, remember, all we need to do is get to minus 55 millivolts. Anything over and above this is simply overkill. So as long as the next pair of voltage-gated channels is close enough so that the first pair can still hit this threshold value of the next pair, then we will see those dominoes fall. If we were to look at the opening of this particular pair of voltage-gated channels, we know that at this point, we will get an action potential that will peak out at plus 30. But if we move away from it, when it's at plus 30 by those channels, we can still hit a threshold difference of at least minus 55 millivolts, that far away from our pair of voltage-gated channels. Think about it as you're throwing a pebble into a still pond. Where it splashes down, you get the biggest wave. That's going to be here, where those channels open and let all those ions cross. The further we get from the place where the stone hit the lake, the smaller the wave we get. Using that analogy, at least at this distance, the wave is still big enough to meet the threshold value for our very next sodium channel, causing it to open. If we were to take these channels and move them further away, the voltage created by this pair of voltage-gated channels now may not extend far enough down the axon to meet the threshold value of the next pair. So they do have to be close enough together. Be no different than if we had our dominoes set up again and we didn't put them close enough together. You'd knock down the first one, it would topple over, and it wouldn't reach the next pair. So they do have to be close enough together for this to happen. And by looking at how the voltage changes with each particular pair of voltage-gated channels, we see this propagation all the way down the axon. Now, we discussed Schwann cells and oligodendrocytes last session. We talked about how they are associated with the axons and they help the transmission occur more quickly. Not all axons had these Schwann cells associated with them. So because we can have situations where they're present and where they're absent, we are going to have two slightly different mechanisms on how these transmissions get down the axon. One is the continuous conduction. Now, unbeknownst to you, that's what we've just been discussing. Propagation along an unmyelinated axon. No Schwann cells, no myelin. All we have was bare axon and our pairs of voltage-gated channels spread out, equidistant, but close enough together so that the electrical effects of one could still meet the threshold value of the subsequent group. The next type is saltatory conduction, and that's where we have our Schwann cells associated. This is going to be the faster transmission, and as you'll see some resources describe it, that impulse will hop or jump from node to node to node. Now, I don't encourage that analogy, but I put it in here because you are going to see it come up in other resources. But please do not describe this as a jumping effect, because it's actually the opposite way I would like you to look at it. When we have myelin present, we get a faster transmission because those Schwann cells and that myelin sheath insulates the axon to any unwanted transport. As we said, in a unmyelinated axon, we had all those channels spread out evenly along the length. In a myelinated axon, the only place we see them is at the nodes. The spaces in between our Schwann cells, where we happen to have bare axon. So we would see clusters of those voltage-gated channels at each of these nodes of Ranvier. And we know the Schwann cell has an insulating quality, and that's how it speeds things up, because we simply don't see any channels underneath the Schwann cells. They'd be pointless. They would just cover them up anyway and block the openings to allow things to enter and leave. Anywhere where you see a Schwann cell is an area of that axon that is unavailable to transport. And that's important for quicker transmission. Remember, when we looked at an unmyelinated scenario, when this sodium comes in, it gives us that vast depolarization that then allows our second sodium channel to meet its threshold, open up. So it creates its own depolarization and action potential, which is large enough to open up the next one, and so on and so on and so on. But in order for us to still have an electrical change of that magnitude, we still have to have that sodium kind of lingering in here. If we have all of this axon available to transport, we are going to start to see that sodium simply leave the cell. And it's going to dissipate any electrical effect it would have had. It basically decreases the reach that this sodium channel would have in trying to meet the threshold further away from its central point. But if we put a Schwann cell here, let's draw the Schwann cell in red. If we put a Schwann cell here, now we had all that sodium entering the cell. But we've got this Schwann cell insulating that part of the axon now. With that Schwann cell here, the sodium that just entered the cell really has nowhere to go. And as long as it stays inside the cell, we will continue to get a depolarization. Distinctly enough, a depolarization that's big enough to meet the threshold value of our next voltage-gated channel. Now, this is not an indefinite phenomenon. We couldn't simply have one big Schwann cell covering the axon and call it a day. There are still limitations to how that electrical event can reach down the axon. But the presence of the Schwann cell helps a great deal. And it allows us to take the distance that we normally had in between one pair of voltage gated channels and the next and spread it out. Because we know that when the Schwann cell is present, the sodium that enters here can have an effect, at least to meet the threshold value, much, much further along. Where normally, if we look at this diagram now, without the Schwann cells, we may have needed to have another pair of voltage-gated channels here, and another pair here, and another pair here. But because we've insulated this entire section of axon using the Schwann cell, any sodium that enters has nowhere to go anyway. So we can still measure an electrical effect at least to the degree of minus 55 millivolts, all the way down to this portion of the axon. Now this becomes a faster event because we didn't have to take time to open this pair, or this pair in sequence. Now granted, we're only talking about a fraction of a microsecond really, but when we look at the length of an axon, that all adds up. Fewer steps. So rather than picture that we've increased the speed of transmission, what we really did was functionally shorten the axon. So we're able to get from the axon hillock down to the axon terminal in less amounts of time. Now this diagram also is a good indication of why I hate the analogy of an impulse hopping from node to node. Even this diagram has an arcing arrow implying that. But if you are thinking about describing this as a hop, really what you're doing is focusing out here. I want you focused in here. This is where the electrical activity changes and the reach is extended down the axon because of the presence of our Schwann cell. So please don't describe it as a hop. If ever asked how the impulse travels more quickly down a myelinated axon, your answer has to involve the insulating effect of those Schwann cells. The Schwann cells will insulate that portion of axon against any ions fluxing back and forth. So therefore, when we open up our clusters of voltage-gated channels at the nodes, the great electrical effect that we get because of that has a greater reach down the axon. So we can start opening up pairs of voltage-gated channels further away. So now, what would happen if we had a myelin sheath problem? What about if we had a myelinated axon that's all of a sudden no longer myelinated, or parts of the myelin missing? We see that, I don't want to say often, but we see it with certain diseases. Multiple sclerosis being probably the most popular or common of these demyelinating diseases, where for one reason or another, the Schwann cells, or actually in this case, it's usually oligodendrocytes, because we will see demyelination in MS occur at the brain stem and the brain. No Schwann cells there. Those are where the oligodendrocytes are going to hang out. And because now where the voltage-gated channels happen to be, the impulse will start at the axon hillock, continue down the axon, reach the place where we're missing the Schwann cells, and stop. Now, what symptoms would we expect to see in a case like that? Remember the controlling nature that these nerves have. We're going to rely on this nerve transmission for sensation. This same nerve transmission that we're discussing now would be used in a sensory manner. You prick a finger, The receptors that are stimulated there will extend up the arm and into the brain. That's electrical transmission. We rely on this transmission for motor function. Something starts in the brain and extends out to a muscle to get muscle contraction. If we can't get the transmission to the muscle, we're not going to be able to control that organ. So if we lose this Schwann cell, we lose its insulating qualities. Sodium enters here now, and without that insulation, it starts to leave the cell again. The more sodium that leaves the cell and goes back outside, the lower the amplitude of that voltage change is going to be, and it is not going to be at minus 55 millivolts at least to open up the next pair. So the transmission would come to here, we'd get an action potential, but it would not be enough to carry through this part of the axon, and that's where it would stop. So we would get muscle weakness. We would get areas of numbness. If it's a nerve like the optic nerve, we may get loss of vision. And for MS in particular, we'll see these demyelinating plaques where we start to lose our oligodendrocyte myelin sheath. This one's rather dramatic. This is the corpus callosum. You would have seen this in the sheep brain in lab. Large bands of axons connecting the left hemisphere to the right hemisphere of the brain. Look how deteriorated it happens to be in this particular scenario. This darkened area here is the ventricles in the brain, the lateral ventricles, containing cerebrospinal fluid. Here would be a normal pair. Look how they're opened up as the white matter containing those oligodendrocytes has deteriorated and compacted. So that's propagation down an axon. Now, hopefully already, somebody here has been thinking ahead. Because we said that this sodium opens up this potassium and this sodium. And this sodium opens up this potassium and this sodium. That's how we march our way down the axon, right? But what prevents this sodium from opening up this sodium? Surely, if this one here opening, if I can get my marker working, if this one opening can open up this one, the distance is the same for that second one now to go back and reopen up that one. But we don't see that. We only ever see this transmission march from the axon hillock down to the axon terminal. We don't see it propagate the other way. And it's important that we do control this. Otherwise, you start off with an action potential at the axon hillock, and then after that, all we're going to get is this reverberation back and forth the axon of random voltage-gated channels opening and closing. We want to have a scenario where we can start at one end, send an impulse to the other, and then that event as an entity is done. Then we can start with a second impulse if we want to. But we always want to maintain the ability to have one directional flow. We accomplish that, or it's accomplished, because of two different things. The first is simply the characteristics of the channels we've been talking about. Remember the sodium voltage-gated channel had certain characteristics. It opens up at minus 55 millivolts. It closes at plus 30 millivolts. And when it closes, an inactivation protein seals off that channel. And then there's a period in time where it cannot be opened again. The potassium voltage-gated channel would open at plus 30. would begin to close down around minus 70 millivolts, but it's a slow closing. And we get excess potassium leave the cell, and we get this hyperpolarization period. Those are the characteristics that help encourage one-directional flow. Specifically, the inactivation period of the sodium, the hyperpolarization period of the potassium, and the nature of these two channels essentially competing with each other. Now, what do I mean by that? We said that this sodium here opens up its partner potassium and the next sodium. So essentially, this potassium and this sodium opens up at the same time, right? Or at least near enough makes no difference. It's the sodium rushing into the second channel that gives us our depolarizing effect, right? It's the one that opens up the next sodium, which then opens up the next sodium. But we don't get the same effect in what we call the retrograde direction, the reverse direction. Because as sodium comes in and then spreads out up and down the axon, down the axon, we're bringing in all those positive ions in an undisturbed axon. Our positive and negative poles are exactly the way we want them to be in a resting state. So these positive charged ions that are entering has a great deal of electrical impact in this direction, in the anterograde direction. However, look what happens to the sodium that's entering and it goes backwards. It's a positively charged ion entering the cell. Look what other positively charged ions in the same vicinity has left the cell. It doesn't matter that one's sodium and one's potassium. They are both positively charged ions. Electrically, they're going to cancel each other out. So these positively charged ions are heading backwards in this direction to the previous pair of voltage gated channels that have been opened. But this is by any stretch of the imagination undisturbed axon electrically. These sodiums are electrically competing with the potassiums that have left the cell, canceling its effect out. So I had drawn a circle earlier where I looked at this particular sodium, and I described this as like a circumference of influence. That when sodium comes in, this is how far away the effects of that sodium can still open up voltage-gated channels if they're close enough. And if that particular axon was completely undisturbed in either direction, that would be true. But of course, we know that's not going to be the case. We would have always had this pair open up just before this pair. And because of that, that quote-unquote circumference of influence now is skewed. The electrical effect from the point in which the sodium enters is much greater in this direction than it is in this direction. Because the loss of the potassiums through that previous channel cancels out the electrical effects of the sodium that just entered through the subsequent channel. So the nature of the channels and the nature of the ions that are fluxing in is the first reason that we get one-way propagation. The next is the refractory periods. And the good news is this is not like a whole new topic now, because these refractory periods are also a factor of the characteristics of the channels that we just discussed. These refractory periods are two periods in time. The absolute refractory period is the period in time which we absolutely can no way, it's impossible to get a second action potential. So we kick off an action potential here. It is impossible to get another action potential even started until we at least reach this point in time. Because remember, all this is time, right? This is a sequence of events. The sodium channel's opening, the potassium channel's opening, the sequence of events. Remember, the sodium channels opened, they closed, and then they went into an inactive state. The end of the absolute refractory period marks the end of that inactivation state. That's when the sodium channels at least reset back to normal, and it's possible to reopen. So you have at least the possibility of a second action potential. The yellow shaded area is the relative refractory period. During the relative refractory period, we can get a second action potential, but we need a larger stimulus. Now, it's that word stimulus that's going to cause some of you headaches. Remember, we're putting all of these into an electrical context. How did we get this particular action potential started in the first place? If you don't know that, go back and watch the previous lecture again. We needed enough chemically gated channels to open to cause a graded potential that was large enough to hit the threshold value of our sodium voltage gated channels. The number of chemically gated channels open and the amount of sodium that entered, that is relative to the stimulus. A bigger stimulus on a neuron essentially means it got bombarded by other neurons. More chemically gated channels were open at the same time. More sodium fluxed in at the same time. And a greater electrical change occurred. In an undisturbed neuron, we had to go from minus 70 to minus 55 millivolts. We had to let in enough sodium to change the membrane potential by 15 millivolts. But look where we are for this relative refractory period. We're never at minus 70 millivolts. This entire relative refractory period coincides with the hyperpolarization phase. And anywhere along that period of time, we're going to be further away from threshold than we would have been at rest. At any point in that period of time, we not only have to let in enough sodium to go from resting membrane potential up to threshold, but we also have to make up this gap, this hole that we've dug ourselves in, because we've hyperpolarized the membrane. This is essentially what we would have seen in the last lecture when we talked about inhibition. We've got an inhibitory effect happening during our relative refractory period, making it more difficult to reach threshold and get a second action potential. But it's still possible. With a large enough stimulus, we still can get another action potential occur when we're in that period. Now, how does that reinforce one directional flow? Time. Time. This relative refractory period and the absolute refractory period buys us enough time. So if we go and look at our domino effect again, where this pair opens up this pair, which opens up this pair, Well, remember, when this pair opens, now the sodium channel is going to go inactive. While it's inactive, this pair has opened, which has opened up this pair, which has opened up this pair, which has opened up this pair. Okay, now this sodium is ready to roll again. But look how far down the axon we've traveled. There's no way this pair or this sodium channel is going to have enough of an influence to open up another sodium channel that far retrograde or backwards on the axon. So that inactivation period in our sodium channels, reinforced and supported by the hyperpolarization phase in our relative refractory period, buys us enough time that any electrical events that are happening in that axon now are far enough down the axon that they cannot affect us this far back. So that's how we encourage one directional flow. The nature of the ions going back and forth the membrane, with the sodium entering at one pair of channels, competing with the potassium that has left previously, and the nature of those channels giving us our absolute and our relative refractory periods. The absolute refractory period is directly due to the inactivation period of the sodium channels, while the relative refractory period is directly linked to the hyperpolarization phase caused by our potassium channels. So now that impulse has extended down the axon. It's finally made it to the end. Now we've got to take some mechanism and ensure that all that effort has allowed that neuron to influence whatever's on the other end of the axon. Now, whatever's on the other end of that axon could be another neuron. Remember, we talked about presynaptic and postsynaptic neurons in the last lecture. If you have two neurons and one's influencing another, then one's going to be the presynaptic neuron and one's going to be the postsynaptic neuron, always because of that one directional flow. But we may be looking to use that impulse to recruit an organ. If it's two neurons, we call it a synapse, and the space in between is a synaptic cleft. And this is where we're going to be coming full circle now on the material that we started with. Impulse comes down the axon, pairs of sodium and potassium voltage gated channels. Well, at the end of the axon, we don't have sodium and potassium voltage gated channels anymore. We've got calcium voltage gated channels. But they open same way that the sodiums did with a threshold. They're still a voltage gated channel. But they let calcium into the axon terminal. When calcium enters the axon terminal, these vesicles in the axon terminal storing our neurotransmitter merges with the end of the axon membrane and dumps its neurotransmitter into the synaptic cleft. Now, when we started talking about this, we would have simply called these a chemical. The channels they bind to here on the other side of that synaptic cleft, we would have called chemically gated channels. So this is the chemically gated channels in a neuron, with these neurotransmitters now being the chemical that opens them. Sodium rushes in. Now we got a brand new graded potential in this neuron. So we've kind of come full circle. The events we just discussed here were the same events we discussed previously when we talked about how do we get an action potential to start with. Calcium entering the end of the axon, triggering the release of those neurotransmitters. Now, the neurotransmitters in that last diagram was acetylcholine. There's any number of neurotransmitters, depending on the nature of the neuron and what it's trying to accomplish. Think about the neurotransmitter as a chemical messenger. It is the specific chemical that opens up a channel on either the next neuron or the organ. And because of that, we can have a geographic description of a presynaptic neuron and a postsynaptic neuron. Now, this is the term that we brought up last class. Here's neuron number 1. Here's neuron number 2. This is a synapse. So this would be the presynaptic neuron, and this would be the postsynaptic neuron, because that electrical impulse only moves in one direction. We used that term in the last lecture when we used the descriptors for our different types of graded potentials. We had an excitatory postsynaptic potential and an inhibitory postsynaptic potential. Both of those were describing graded potentials at this neuron, the postsynaptic neuron. If the neurotransmitters from the presynaptic neuron opened up channels that caused the voltage in our postsynaptic neuron to decrease, then that was an inhibitory postsynaptic potential. If the neurotransmitters released from the presynaptic neuron caused the electrical activity in the postsynaptic neuron to elevate, depolarize, reach threshold, now the graded potential that we're seeing here is an excitatory postsynaptic potential. Now, there's any number of different types of synapses. And really, there isn't enough time in this class to get into each of these and their relative functions. For now, just picture the generic types. We have an axodendritic synapse, where we get our axon terminals from one neuron innervating the chemically gated channels on the dendrites of a postsynaptic neuron, or an axosomatic synapse. Other types of synapses have completely different functions. That's a topic for another time. We see various arrangements of these synapses. The converging circuit is a rather common one, where you can have, at least in this diagram, 4 presynaptic neurons, 1 single postsynaptic neuron. And remember something that we said before, these two may be inhibitory. These two may be secreting neurotransmitters that are excitatory. Whichever one is sending the impulse is the pathway that's going to dictate if we get an action potential in our postsynaptic neuron or not. So that converging circuit is rather common. The diverging circuit is not. And I swear they only include a diverging circuit because they included a converging circuit and they felt like they needed to do the opposite shape. But you should probably appreciate, without me even telling you, that the amount of neurotransmitter we could secrete from the end of one axon is not going to have a huge electrical effect on 3 different neurons at the same time. There simply isn't a dose effect large enough for that to have an impact. So to expect one neuron to somehow bring about this cascade of events where you get 5 neurons affected at the end, that's a stretch. The reverberating circuit, however, we have use for. Think about this as almost like a feedback loop. Now, don't get that confused with positive or negative feedback, because that's not what we have here, because there is no control center. But if we're sending an excitatory signal down this neuron, we may want to have a system set up where we get an inhibitory signal sent back to turn it off. Neurons need 2 things to stay alive: nourishment, glucose and oxygen, but it also needs activation. So sometimes we'll see these reverberating circuits. This will be the functional portion, but we will get a signal sent back to innervate the earlier neurons to keep them activated, to keep them nourished, to keep them excited. Now, these are also the kind of circuits that can be acquired over time. Trauma, concussions, degenerative states, Parkinson's, Alzheimer's, dementia, stroke. You may get damage to the brain and a subsequent rewiring that is not always beneficial. This is the sort of phenomena we acquire when we get chronic pain. Think about someone who has a devastating ankle injury that gives them a very strong, painful stimulus for a very long period of time. And that stimulus is being bombarded into the brain, lighting up areas in the brain responsible for pain perception. Well, if we get a dominant pathway like that, we do get new synapses laid down. Now that same person with the ankle injury, maybe their therapy didn't go so well, and it was deemed that they need an amputation. So they amputate at the knee. That person may still have what they perceive as ankle pain. You've heard of phantom limb pain, right? Yet the ankle isn't even there anymore. There's no longer a receptor being stimulated by an inflammatory process. And now that pain they're feeling is purely a central nervous system phenomena. Right? You've heard people say, oh, pain is all in your head, right? Well, chronic pain, that's the only place it can be. It's this self-perpetuating circuit stimulating parts of the brain that perceive pain. And then lastly on this particular slide, we have our parallel circuit. Now think about this as a self-protective mechanism. You don't want to put all your eggs in one basket. So you send multiple pathways from the same origination point to the same destination. So trauma, degeneration, whatever the case may be, you lose one or two pathways, you may still have a means of innervating the neuron or the organ at the end. So this was a rather complicated topic that we just went through. It involves understanding how ions change the membrane potential of a cell. It would have worked to your advantage to truly understand the previous lecture before attempting this one. Understand how we generate an action potential first. Then you can better understand how that action potential self-propagates down the axon and spreads. So you should understand the nature of the channels and the ions that are at work when we propagate an action potential down the axon. Saltatory versus continuous conduction. The importance of the refractory periods. And finally, what happens at the synapse. And being able to describe the synapse in the terms that we just did. Presynaptic, postsynaptic. So as usual, if you have any questions, let me know. This here now marks the end of our most complicated material. And it's not even close. Discussions of neurons, action potentials, greater potentials is by far the most technical material we will get through in this class. Everything else is something that people can wrap their heads around so much more easily. So take advantage of any opportunity you have to discuss this particular topic with classmates, tutors, spotlight sessions by other instructors. Because if you can get past this particular topic, it's somewhat smooth sailing after this. So take care, good luck, and we'll meet again to continue on with the rest of our nervous system discussion.